Briefly about this yesterday, and today is the 18th. Okay. So I think this is where we left our heroes. Right there. What fossils show and what they don't? Remember this slide? Does this look familiar? You were on the slide before that? Okay, then your quiz is going to be out of 4, not or 3, not 5. Cool. Or 4, not 5. I'll look at it. Okay. That explains why a lot of you got a lower score than I would have expected otherwise. Huh? On the last slide. Okay. Well, let's go to here then.
Yeah.
Okay, so you guys actually weren't even ready for that quiz. I apologize.
Well, if we're up for it anyway.
Yeah. All right, so starting all over, today is the 18th. So we said that fossils are either actual pieces of organisms or impressions left by them. You can have bones that fossilize. You can have whole insects that are trapped in pine sap, and that gives you amber. Fossils, for the most part, I mean, the critical piece is an organism has to get buried in something. But not all things you can get buried in give you an equal likelihood of producing fossils. Moist environments with very fine sediment tend to give you your best fossils. So muddy, swampy areas, shallow oceans, volcanic areas with ash give you great fossils. Stream edges can give you pretty good fossils. And you can actually, what's nice is the edges of streams today, very often you can find old rocks that have been exposed as the stream cuts into older rock layers. So like walking along the edge of Beaver Creek is a great place to look for fossils. These are all good places to find fossils. Notice that not every place on the planet is either a swamp, a shallow lake, a stream, or a volcanic area. There are a lot of places on the planet that don't produce good fossils. Okay. Fossils don't show everything. What fossils do tend to show? Hard-bodied organisms, or those with skeletons, and woody plants, so more tree-like things. Fossils they show a geologic time of death. They don't usually show soft-bodied stuff. You don't get worm fossils. You don't see a lot of fossil organisms from grasslands, from deserts, from mountains, because things tend to decompose there and not leave fossils. Okay, so would our raccoon, the raccoon that we said might have died in mud yesterday, could that form a fossil? Yeah, easily. Anything with a skeleton tends to form good fossils if it dies under the right conditions. Bacteria don't leave good fossils. There are a few rare exceptions to that. The stromatolites we talked about a little bit are an exception. Earthworms, jellyfish don't leave good fossils. Anything like that tends to not leave fossils. When you have fossils in a rock, you can calculate the geologic age of the rock, and that's using radiocarbon data, or radioactive dating, radiometric dating. Then you can figure out how old those fossils are and arrange them by age. The other cool part is that rock layers form with new layers on top of older ones. This is called the law of superposition. And geologists, it's an art and a science. They can look at a rock layer, no matter how folded up and banged up and bent and flipped over it's gotten, and they can figure out how old that rock layer is. So, and they know that if this is a 3.2 million year old rock layer, I know that doesn't look much like old, then whatever's above it is younger, because you have newer rock on top of older rock. And then when we look at those fossils in the rock, once we line them up by age, we can see which ones came first, and we can generally see those evolutionary stages, that sort of accumulation of small changes that lead to big changes. in time. So again, once we know how old the rock is, we can establish how old the fossils are inside the rock. And there are some, like I said, there are some fossils that are so specific to a particular era that when we see those fossils, we know, oh, that's how old the rock is. Then we can confirm it with radioactive dating.
How do they look at a fossil of, like, obviously something that's not modern and know what it is?
That's a great question. That's a fantastic question. So if you're an anthropologist or a paleontologist and you unearth a fossil, there is an entire science behind measuring portions of that fossil. So you look at overall structures and you look for things. So here's the hard part. Are people done with the slide? It's a great question. Recording. Because we've got so many fossils of so many species from all over the planet that we've been collecting for the last couple hundred years. I mean, realize that like dinosaur fossils were being found in Europe in the Middle Ages. Do you know what they thought they were? Dragons. Dragons. These are the bones of dragons. These are the teeth of dragons. Well, it makes perfect sense, right? I mean, you find these big giant bones, they must belong to some sort of monsters and they must be around here somewhere, you know. So we've been collecting fossils for a very long time. And just like biologists look at structures and shapes to sort of classify organisms, we can look at two living things and say, okay, there are similarities here, we think they're related. Just like I can look at some of you and your siblings and say, I bet they're related. We can also, now when you're starting from scratch and you have one isolated fossil from 3 million years ago and you pull it out of the ground, if you don't have anything to compare it to, it may not look like anything modern. And then you're like, oh, dragon. But when you've got this huge sort of library of fossils, and when we go to the Carnegie, so when I was a kid, the Carnegie was more like what I'll call the old-fashioned Natural History Museum, just rows and rows and rows of skeletons. Cool! I thought it was the coolest place on earth. They have those things back in their collections. So they have rooms where researchers work with very old fossils, very old skeletons. When you've got a whole library of things, you can look at a new fossil and go, okay, yeah, it doesn't look like anything modern, but look, it kind of looks like this, but a little bit different. Oh, and it kind of looks like this, but a little bit different. Because for the most part at this point, in terms of the fossil record, it's kind of like if we take, so if you imagine reading a book, and if I give you pages 1 through 6, and then page 8 through 10, and then you randomly find a page, you can probably figure out whether it fits between page 6 and page 8 or not. If I give you page 6 and page 93, without page numbers, you probably can't tell where these fit. So at this point, we've got a lot of the pages, and every here and there in the fossil record, it's like we're missing a page or two, but we don't have giant gaps for a lot of species. So it's not as hard as you would think, because we've been collecting fossils for... Humans are collectors. Have you noticed this about us? We tend to pick up cool shiny things and take them home and look at them, whether it's rocks or sticks or bones or plants or whatever. And biology and paleontology and anthropology is a lot about collecting stuff to compare it to other stuff. And museums and universities usually house these enormous collections of fossils. When we go to the Carnegie, there are still areas where they've got some fantastic examples of that sort of progression of fossils. Well, if you found a fossil, you could easily fit it in between two other ones. It's not like a random page and you have no idea where it's coming from or where it's going. Does that answer the question? Mm-hmm. Okay, cool. Okay, so we've got this amazing fossil record. Like I said, it doesn't show everything, and it doesn't show all species, and we do have some gaps in it. But when we find new fossils, it's not that hard to sort of plug them in. That's the first line of evidence for biological evolution. The first line of evidence that we deal with is fossils and the fossil record. I'm just writing fossils, fossils. I don't know why I'm doing that. So we'll call that the first line of evidence, fossil record. The second line of evidence is structural evidence. And I think the video called this sort of comparative anatomy and physiology, or comparative anatomy, basically looking at similarities and differences in the structure of living things. And your biology book addresses it as structural, and I like talking about it as structural because we're talking about structures, and you all know what structures are. So the first kind of structural evidence we'll talk about, homo means same. Homo means same. Yes, homosexual is the first thing some of you thought of. That's fine. Homosexual means two people who are the same biological sex.
Well, I said the wrong thing. I said one.
Oh, okay. Somebody else said that. That's so funny. Somebody in the other class said one. It means same. Hetero means different. So heterosexual is two people of different sexes. Homosexual is two people of the same sex. Homologous structures, homologous is how we usually pronounce it, are structures that are the same in some way. They are analogs of one another. They're like an analogy in bone. So they're structures that appear in different species but share a shape. Homologous structures show common ancestry. And the classic example is the bones of the human arm, the bat wing, the whale flipper, and the cat forelimb. So homologous structures are structures in different species. This is really important. And you're going to have a probably 3-question recall quiz. The way I'm going to do this, I'm going to post this after school today. You can take it on any computer you want. I would advise, I only am posting it after school because, well, I might have time 7th period. Check right at the end of the day. I would like you to do this while it's still fresh. So I don't really want you to come in Monday morning having not thought about any of this in 2 days and take the recall quiz. So it's going to be a recall quiz that's on the stuff we've done, and I will have it up by the end of 7th. How about I just promise you that it'll be up by 2:45. And I would like you to take it. The big thing to understand about the homologous structures is these are in different species. These are not in the same species. We're comparing structures in multiple related species. And if we look at the arm bones example, so in humans, cats, whales, bats, dogs, deer, pretty much as far as I know, all mammal species, you have a single bone, 2 bones, a bunch of little bones, and then these long phalanges type bones. Every single one. Oh, hey, look at the cat. Single bone, double bones, bunch of little bones, long phalanges type bones. Huh. In the whale, single bone, 2 bones, bunch of little bones, very long phalanges bones. And in the bat, same thing. Single bone, 2 bones, long phalanges type bones. All mammal species have that same arrangement. Now, at some point, is it possible that it will not be worth the extra energy to grow 2 bones, and whales would be my candidate, that some whales in 1,000,000 years will have 1 bone there? Is it possible that in 1,000,000 years, and descendants of whales will not spend the energy growing 2 bones? Yeah, it is. Could whales in 1,000,000 years only have 1 bone here? Yeah, they could. That's evolution.
Okay.
But I betcha
All right, thank you.
that if in 1,000,000 years whale descendants only have 1 bone there, when you went back to looking at whale embryos, you would see evidence that it had started off as 2 bones. Do you remember the dolphin in the video? The little dolphin. What's an embryo, first of all?
An unborn.
Yeah, it's a little unborn creature of some sort. So you can, anybody here have chickens at home? Who has chickens? I love chickens. So do you have roosters also? Do you get fur legs? No. Okay. So if you've got roosters and chickens, roosters do what roosters do and chickens do what chickens do, and you get fur legs. And that's an egg that has an embryo in it. And if you look at those chick embryos, you can compare features. The study of embryos, do you remember what it's called? Embryology. Yeah. Ology being the study of something. So I would bet in the case of the dolphin, do you remember where those nasal slits started out on the dolphin? Started out on the front, and they actually migrate up the skull during development. I bet you in 1,000,000 years, if whales only have 1 bone, when you looked at whale embryos, you would see development, starting off with 2 bones and fusing. It's possible. And it's also possible that that doesn't happen because, you know, I'm conjecturing here. So that's the first kind of structural evidence, is homologous structures. The next kind of structural evidence for evolution is what we call vestigial structures. Vestigial structures are structures present on modern organisms that serve no known purpose. Vestigial structures are often reduced in size. They are considered evidence of an organism's evolutionary past. Examples include snake legs, what? And whale legs.